webinar series. It is my pleasure to chair these sessions that will take place once a month until the end of 2020. First of all, I hope that all of you are safe and far away from coronavirus. We are going through strange times, and we have been forced to adapt our lifestyles and working routines. I would like to introduce NANOCELIA Network to you. NANOCELIA started in 2017, and it is an Ibero-American network funded by FIDET. Its main aim is to boost the nanocellulose research developed in Ibero-American countries to companies, promoting the transition from the lab to the large scale in production, characterization, and application of nanocellulose. At the beginning, we were few members from different countries, but the network is becoming bigger and bigger, and we are really happy of that. If you want to stay tuned to all the breakthroughs and news from the network, do not hesitate to contact us. I will write the contact details below, as well as the website of the network, where you will be able to see the different activities, members, and description of the network. Well, we will start our webinar series with Dr. Javier Carvajal, Director and Principal Investigator from Universidad Pontificia Católica del Ecuador and at Neotropical Center of Biomass Research, Professor at the Department of Biological Sciences and Innovation CEO at Saccharis. The topic of his presentation is the tagua-based biorefinery, a nanocellulose and other natural products platform. At the end of his presentation, there will be time for questions and discussion. So please, use the chat at the right side of your screen to ask as many questions as you want, and I will formulate to them. So, let's start with the first presentation of our NANOCELIA webinar. So, Dr. Carvajal, the screen is yours.
Thank you very much, Mark, for your introduction. Today I will present this work that we have been performing in the Neotropical Center for the Biomass Research since the last 10 years. The title is the tagua-based biorefinery, nanocellulose and other natural products. I have to say that this series of presentations, virtual presentations, are sponsored by NANOCELIA Network and CITED. I am a professor at the Pontifical Catholic University of Ecuador, and well, my name is Javier Carvajal. First, I want to show you the contents of this presentation. I will introduce my center, Neotropical Center for the Biomass Research. Then I will move to the biorefinery concept, basic concepts of biorefinery, the tagua approach. Then I will talk about the biorefinery platform and the industrial scope, nanocellulose from tagua, the worldwide sources and economic potential for other seeds similar to tagua, and finally I will conclude. Well, the Neotropical Center for the Biomass Research was founded by 2008 in the Catholic University in Quito, and I will just show a little bit of what we do in the center. We work pretty much on natural sources from Ecuador. Ecuador is a biodiverse country, so we have many different sources in microbial, animal, and vegetal species. We can visit this web page, but let me show you the most important parts of our center. First of all, we have the yeast collection, where we preserve more than 3,500 yeast isolates from all over the country, including the Galapagos Islands. The other important part of the center is dedicated to nanoscience. So we work pretty much on the research of new nanoparticles, bio nanoparticles, especially from natural sources in Ecuador. We also have a biorefinery pilot plant. This is a photograph of our pilot plant. It was built between 2008 and 2012. We can produce here alcohol, fuel alcohol, second generation fuel alcohol, as well as fertilizers and other products. And here you can see the facilities we have in the biorefinery, and this is the place where we scale up our work of research from the labs. And finally, we tend to the techno-based enterprises, and here you can see what we did with nanotechnology. A few years ago, we were able to resuscitate some yeast species from the oldest brewery of the Americas, founded in Quito in 1566. So afterwards, we rescued the original recipe, and this is a product that came out from our research in yeast and nanotechnology. We have also started a new project on bioethanol production for the university. This is an alcohol gel that we produce in our biorefinery. So I invite you to visit this webpage so you can write us, and if you have any questions, we will be very glad to respond them. Okay, so the biorefinery concept, for those who are not familiar with this concept, we have the traditional facilities for extraction, for example, sugarcane as the initial biomass. Normally, they extract the sugarcane for sugar, and then they do not produce anything else except alcohol or sugar. But there is another way to think about the leftovers and the residues. In this case, in a biorefinery concept, the sugar extractions goes to the sugar process, but there is also sugar cane bagasse that can be hydrolyzed to obtain cellulose and hemicellulose. This hemicellulose and cellulose can be converted into second generation ethanol for fuel for moving engines. In the fermentation process, yeast produce CO2 that can be used to enrich algae culture medium, and this algae biomass is very important for industries such as cosmetics and food and feed. So CO2 is fixed into biomass again. And finally, we have wastewater that through an aerobic digestion can be converted into biogas and biofertilizers that can be used in the same process. Biogas for moving engines and biofertilizers to be used in the sugarcane crops. That's the perfect way of closing the loop, and that's the general concept of a biorefinery. In our case, we have developed a different concept of biorefinery with tagua. Let me show you, sorry, a little bit about tagua. Most people does not know this palm tree. The geographic distribution of Phytelephas equatorialis, the name of the species, goes from 0 to 1,500 meters above the sea level in Ecuador, in the Pacific coast. Ecuador is in between Colombia and Peru and South America, in the northwest South America. We have 4 natural regions: the Amazonia, the Andean region, the Pacific coast, and over there in the Pacific, we have the Galapagos Islands as well. So, as you can see here, we have, even in the shores of the Andean region, we have some populations of these Phytelephas equatorialis. I have to remark that this species is not industrially being exploited. Every palm grows naturally, is wild, but there is enough palms to produce buttons, for example. Ecuador exports buttons made out of tagua, and because of this, the special characteristics, physical characteristics of this material that makes it very strong and bright and very easy to work on the cravings and things like that. So let me show you the fruit of this palm tree. I know this is very strange fruit. Almost no one can understand or can believe that this is a fruit. In fact, it looks very much like an extraterrestrial fruit. Every single bunch of this has about 10 to 12 small fruits, and every single fruit has inside from 6 to 7 seeds. The mesocarp is food for small rodents in the forest, and these rodents are in charge of the dispersion of the seeds. And that's how this palm tree can populate different zones in the forest. We are interested in this part, in the endosperm of the seed. As you can see, this is whitish and this is bright, resembling ivory. That's why this species is also known as the ivory nut palm tree. I want to show you, I want to take you to a travel from the biggest part, from the geography towards the cell scale and to the nanoscale in terms of tagua. So let me show you first the cells for storage within the endosperm of the seed. And this polarized microscopy of the parenchyma of Phytelephas equatorialis shows hollow cells with very, very thick cell walls. As you can see here in this polarized microscopy, the organization of the molecules can be understood as different types of molecules in different dispositions. So we can see anisotropic and isotropic regions. Another interesting characteristic of these cells is these are very long cells, and hollow cells with a lumen about 20 micrometers. The cell walls are composed mainly of two different molecules. The first one is mannan, and mannan can crystallize in two shapes, different shapes, this platelet-like shape and this fibrillar shape. This one, the platelet-like, is well known as mannan 1, and the second is mannan 2. We are still in the micro scale. We can see the scale bar is 1 micrometer, so you can tell that these are not still not nanoparticles. Another interesting issue or interesting characteristic of this material is that these two different shapes resembles pretty much both starch and cellulose. So we have something in the middle of the starch and the cellulose that we have in higher plants and other vegetables. And all these molecules and crystals are part of the cell wall and are the storage of energy for the embryo when it germinates. As for the cellulose fraction, that is very interesting that even in micro scale, you can see here in micro scale, we can see very isotropic zones. We can tell there is a organization of cellulose in the cell walls. I really like these images because they reveal the organization of the cell wall during the synthesis of it. I have also to remark that all this cellulose is cellulose type 4, because in the endosperm we only find primary cell walls, not secondary cell walls as in other tissues of the plants. Well, the tagua biorefinery platform and the Industries Co. In general terms, this is the this is the general model of fractionation of the tagua seeds that we have developed. We can find in tagua mostly the three important macromolecules: 3 acylglycerides, galactoglucomannan, and cellulose. So that's why within this dotted square, I have enclosed the fractionation core of the technology. From 3 acylglycerides, we can get biodiesel, or we can get food and feed. From galactoglucomannan, we have developed 2 different paths. The saccharification, which is the release of the sugars to produce ethanol or any other product of fermentation by microbial activities or biotechnology. And if we perform a partial hydrolysis, we get oligosaccharides, which are very interesting for prebiotics and other uses in pharmaceuticals. As for the cellulose fraction, in the micro scale, we can add to other polymers to get nanocomposites or simply composites. But if we hydrolyze and functionalize the cellulose, we can get nanocellulose, fibrils, and crystals. And that is the most interesting product that we have gotten so far from tagua seeds. Let me talk about hemicellulose fractionation. Here we can see a unit of the galactoglucomannan that we can find in the seed. As you can see here, mannose is 90% of the polymer, while galactose and glucose is only 2%. Every 200 units of mannose, we can find small branches of galactose and glucose. If we release all the sugars, we can use them as carbon source for microbes. So we can see here the design of our own culture medium for yeast and fungus. On the left, in the upper, in the top, we can see the commercial culture medium. On the right, there is our own culture medium. There is virtually no difference. So we can use that for the design of culture medium, as well as in the other photograph, we can see the fungus growing the same way. That is because both glucose and mannose have pretty much the same transporters in the cell. So for the cell wall or for the cell membrane, it is the same transporting glucose or mannose, and they can enter into the glycolytic pathway and they can get energy the same way. Well, as I told you before, we can produce ethanol. Our experiments showed that when we released the sugars, mannose, we can tell this very conspicuous peak of mannose. After a few days, it disappears and appears glycerol and ethanol, and also a little lactic acid. That means that tagua is also a very good source for the production of second-generation ethanol. And that's part of our biorefinery platform. If we don't release all the sugars, but also only perform a partial hydrolysis, we can get a collection of oligosaccharides, including mannose, glucose, manobiose, manotriose, and manodextrin. This fraction is very interesting and very valuable in the market. Well, so let's talk about nanocellulose fibers and crystals. This is the kind of fibrils that we can get from tagua seeds. As you can tell, they can bend, they are very long, approximately 1 micrometer or more, even more, 1 micrometer length or 1.5 micrometers length, but they are very thin, from 1 to 5 micrometers width. That makes, and they are also very uniform. In this case, I'm showing you the nanocrystals that we got from tagua, and again, we have very similar sizes, from 200 micrometers to 300 and even 600 nanometers, sorry. But all of them are extremely uniform in terms of width. So this makes a very interesting material, strengthening material for composites, while the fibers can be used, for example, for trapping ions, molecules, for filters, etc. Here we have some of the applications of our nanocellulose. On the top left, there is the hydrogel obtained from our process. This hydrogel is quite transparent, and it shows birefringence because the nanocellulose particles are very, very uniform and very thin. Here we can see an aerogel. The pores of this aerogel can reach 20 nanometers of lumen. Here we can see a membrane made of nanocellulose, transparent membrane. On the left bottom, we can see the textile that we have performed in the laboratory. And here in the center, we have this memory-shaped material. It's a composite with nanocellulose. And finally, here we got a photograph of nanocellulose embedded with alcohol. So this is not a hydrogel, but an alcohol gel. And we don't use this for burning. It's only to show that there is no water, but alcohol in there. This alcohol gel is very interesting because it can be used in the treatment of herniated spines and other uses for minimally invasive medicine. Well, I will move to the worldwide sources and economic potential. We have already talked about Phytelephas equatorialis seeds, tagua seeds, but we have also other palm seeds that can be interesting as sources of the fuel, food, and the materials that I have already mentioned. The common features between these seeds are the whitish endosperm, the galactoglucomannan content, the hardness, and the parenchymal cellulose. All of them have the same composition, but in different concentrations. Let's say the Elais guineensis seeds are very rich in oil and much richer than Phytelephas equatorialis and Phoenix dactylifera. So, for example, in the production of oil palm, there is about 6.5 million tons per year of this palm kernel cake. The composition of the palm kernel cake can be seen in this table, where we can see that there is dry matter, protein, crude fiber, including, of course, mannan and cellulose, and other compounds. On the other side, the date production, mostly in the north of Africa and the Middle East, and even California here in the United States, can produce close to 1 million tons per year of this date beets. What does it mean? In terms of business, 6.5 million tons of palm kernels cake can produce nanocellulose, ethanol, oligosaccharides, and oil, and this is a 5 $0.5 billion per year business, and this is a very conservative calculation. We talk about the date seeds, something similar, and due to the lower amount of this seeds, it is $0.5 billion per year. So we can get a lot of nanocellulose, the same kind of nanocellulose as the one as I have already showed you, ethanol, oligosaccharides, and oil. So this is the complete platform of biorefinery. So to conclude, I will first like to talk about the cellular and molecular structure of tagua, which offers very new opportunities for innovation in nanotechnology and biorefinery, because most of the researchers and the industries nowadays are focused very much on the nanocellulose from wood. But the problem with nanocellulose from wood is the intensive energy and chemicals need. To get rid of the lignin, there is a lot of chemicals and energy put in the process. Tagua seeds are free of lignin, and that's a very huge advantage. So that is for sure a new opportunity to develop new materials from different sources in a more sustainable way. And the second point, the fractionation technologies are key to the development of future tagua-based biorefinery. So we have already developed some technologies of fractionation, but still we need much more input from other professionals, including people from business, people from the chemical industry, people from the engineering, chemical engineering, and so on. So the technologies are still to be improved and expanded in this field. It is a really new field and very promising. Third, the nanocelluloses obtained from tagua show high purity and homogeneity, and that is because, as I mentioned before, we extract nanocellulose only from primary cell walls, which produce during the synthesis of these primary cell walls, the cellulose chains are very thin. The cellulose fibers are very thin. So that's why this nanocellulose is highly homogeneous. Well, and finally, a variety of palm kernels can also be exploited under a similar concept. We have been working on different palm kernels during the last 5 to 6 years, including some of the palm trees that we have in the Amazonia, but also from crops. And we succeeded in every single species we tried the same technology. The differences are only dependent on the composition of the quantity of oil, for example, or the quantity of sugars. But all the seeds have mostly the same amount of cellulose, which is between 8 to 10% in the dry weight term. So thank you very much for this opportunity to present you a new source from my country, Ecuador. If you are not familiar with this material, I can tell you that if you visit Ecuador ever, you can go to the crafts market and you can find these small figurines and things like jewelry made out of tagua. Maybe you are wondering why they can paint, why they can stain the seeds, and the reason is because it is a porous material, as I showed you in a previous slide. and so they can make really beautiful works of art with this material. So thank you so much, and if you have any questions, I am ready to respond them.
Thank you, Javier, for this impressive presentation on the possibilities of nanocellulose in the biorefinery, and also the different applications that you have shown. There are several questions that come to my mind, and I will take advantage of my position today to be the first asking to you. So the first point is tagua or ivory nut exclusively found in Ecuador?
No. Fortunately, tagua or ivory nut is the generic name of a series of species. I was talking about Pitelephas equatorialis, but there are many other species, sibling species of tagua, including Pitelephas macrocarpa and others, which are distributed, widely distributed in South America, especially in the Ecuadorian strip of the South America, including Brazil, Colombia, Panama, Peru, and Bolivia. And there are other species in the Amazonia that can also be regarded as future sources for sustainable fuels, feed, food, and materials, of course.
All right, so there is a quite high availability of this resource for the purpose that you have shown today. Yeah.
Yes.
All right. Well, you have also shown, apart from the production of nanocellulose integrated inside the biorefinery process, you have also shown several applications. So are there plans to transfer the technology that you have shown today to the industry?
Yes, of course. In that point, I have to say that we at the university, we are patenting a couple of technologies. One is regarding the production of nanocellulose itself, and the other one is the production of ethanol from ivory nut. In this case, there are also other companies not only from Ecuador, but also from the United States, where I am settled now, and they are interested in these technologies, especially in nanocellulose. So we are in the negotiations, and hopefully in the near future we can do the transfer in fair conditions. That's very important. We have to find fair partners to do this kind of transfer.
All right, I wish you the best in this new adventure, and I hope you will share the main outcomes or the main results with our network, because as you know, this is one of the main objectives of our network, to boost the transfer, or to transfer the research from laboratory to the industry. Yes. Well, and finally, and last but not least, do you consider that ivory nut nanocellulose can become a competitive material in the current markets?
Okay, well, it depends on the kind of industry because, for example, the big paper industry, it does not need this kind of quality of nanocellulose. As I showed during my presentation, our nanocellulose is very homogeneous, very thin, and it is because it comes out from primary cell walls. In the paper industry, for example, they use nanocellulose that is produced or extracted from wood, and they use rather homogeneous sizes, or a more a distribution, a wider distribution of sizes in that case. So I think that the best opportunities for ivory nut cellulose are in the biomedical field and the electronic fields and high technology applications, because it is not only small portion of the nanocellulose that can be produced in the world, but is a very special kind in terms of quality and physical chemical characteristics.
Yeah, both medical and electronics are emerging sectors with a high value added. So, yeah, good luck with this.
Thank
So,
you.
thank you, Dr. Carvajal, again for this presentation. Now we will take a 2 minutes break, and we will be back for more questions. So keep in mind that you can ask anything you want in the chat you have at the right hand of the screen. So thank you again, and see you in a couple of minutes.
Summer is racing fast. Can't you see? There will be no broken hearts here, 'cause I held you real tight in that lingering light. Whispered words that sound so true, and I gave you a kiss saying I won't move from this. You just can't seem to find the words. No, 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 no. I sat on your bed and I watched you sail through silent air. I gave you the world, but somehow world started to fade. I held you real tight in that lingering light, whispered words that sound so true, and I gave you a kiss saying I won't move from this. You just can't seem to find the words. No, 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 no. I pray you don't smile, 'cause I laid down all my guns. Sweet lady, don't you know?
All right. Hello to everyone. Sorry, first of all, for the connection issues that we have had. We have had a problem with the graphic card of the computer, but I think that it is already solved. So I will check all the questions you have made in the chat, and we'll try to move on. So the first question is, what is the efficiency in the process of obtaining ethanol from tagua?
Okay, thank you very much. That's a very good question. The efficiency is exactly the same as with glucose, because as I explained it before, for the yeast cells, glucose and mannose are pretty much the same kind of sugar, due to the fact that the transporters for both sugars are exactly the same. And during glycolysis and then after fermentation, they use exactly the same enzymes. So the time of fermentation and the yield of fermentation is exactly the same.
All right. The next question is if you have ever performed test the performance of these nanocellulose as reinforcing material for composites or other structures.
Yes, of course, we have done that. We have proved some composites by using carbon nanotubes, and we produced a very strong kind of membranes. We also proved mixing the nanocellulose with yeast cell walls, and we got a different property in the membranes. that is flexibility. So we can improve, we can try with many different bio materials, because we are trying not to mix nanocellulose, for example, with other petro-based polymers, but we are trying all the time to develop new biomaterials, new biocomposites. But indeed, nanocellulose, especially crystalline nanocellulose, reinforces a lot the materials. As it's already known for everyone, but this nanocellulose behaves exactly the same as other nanocellulose in that case.
All right, thank you, Javier. There is another question from Professor Luis Ramos. He asks if the exploitation of naturally occurring palm trees is usually a complication. Can this tree be produced intensively? Can you tell us what is known about its agronomic requirements and productivity?
Okay, that's another very good question. Thank you very much. In fact, in Ecuador, there are very, very few crops of tagua palm trees. Still, this species is not domesticated, so it needs a long period of domestication. But they can grow tagua palms in the gardens, for example, in the rural areas. They know that tagua is tagua palm is very interesting in terms of the economy, not only because of the seeds, but also because of the fruit itself. They can eat the fruit, and also the leaves are used for making roofs in the bungalows they used to, they live in bungalows normally. So this is a very interesting species in terms of economy in my country. It is possible to make an intensive crop of that. Still needs a lot of work. Nowadays, tagua produces naturally a lot of palms that are giving enough material for the buttons industry in Ecuador. Every single month, they produce about 400 tons of leftovers. In the buttons factories produce 400 tons. And from these 400 tons, about 7 to 8% is cellulose. The rest, about 70%, is sugars. So you can imagine that we can valorize a residue, not only the primary product, but the residue of the factories.
All right, thank you. There is another question, and it is there any economical advantage on using ivory nut as raw material instead of other lignocellulosic resource?
It depends on the kind of industry that you want to apply the nanocellulose, the final product. I would say that with our technology, we are able to produce nanocellulose of excellent quality in a time period of 5 to 6 hours. So you can imagine that if you compare with the wood nanocellulose, it takes much longer because you have to get rid of lignin. We don't have any lignin in the seeds. And also, we have performed some innovations in our process that we don't need to use other chemicals or tempo, for example, tempo functionalization that is quite polluting to the environment. So we are not only looking for economy, but also we are looking for the environmental sustainability in our processes. I would say that in volume, we have a drawback in terms of the amount of nanocellulose that we can get is much lower compared to the other sources such as wood. But in terms of the quality, in terms of the sustainability, and in terms of the easiness of the production that we have gotten with our technology, we are really competitive.
All right. All right, thank you. So there is another comment that says, if I understand well, you get nanocrystals from primary fibrils. How do you separate primary fibrils from others to obtain nanocrystals?
We are talking about primary cell walls. You have in the tissues, in the vegetable tissues, primary and secondary cell walls. In the seeds, the plant itself only synthesizes the primary cell wall. And the primary cell wall is characterized by this very thin kind of fibers. So that's a natural advantage of the product.
All right, thank you. There is, I think that it is the last question from Carlos Negro from Madrid. The nanocelluloses and nanocrystals that you have obtained are very homogeneous. Which were the production processes and the conditions?
Well, that's that's a very good question, and that's in the process, a patenting process. So it's really well to review all the procedures we have developed there. But I can tell you that we are in a continuous improvement process. We started with the typical acid hydrolysis, but we are moving to other types of processes that we are developing in the labs. So the best answer that I can tell you is that we are really developing a new biorefinery platform, which is completely different to that of used and widely known from wood. In wood platform, you need to get rid of lignin, so you need a process especially for that. You need to bleach the pulp, and you need to get very fine fibers by milling, for example. We don't use that kind of technology. We don't even use right now ultrasonification. We get our nanocellulose crystals and fibers straight forward from our process. So it's a really interesting and new process. Okay, if I well understood, Luis is asking if we can introduce in the process not only the seed but also the husks and on the bunches, and that's the answer is yes, we can do that. But we are talking about second generation ethanol, and this is very important because it gives me the opportunity to launch a new idea. When you talk about first generation ethanol, it is referred to the crops dedicated to that aim. Let's say sugarcane or corn or potatoes or whatever you are using. But in this case, this is not second generation ethanol because we are not using the leftovers of the sugarcane, for example. We are using a different kind of sugars, different kind of feedstock that is not being cropped. It is only collected in nature, and or it is the result of the production of buttons. So I would say that this is a very particular kind of ethanol if we refer to the origin of the raw material from the seeds. It's different. And if we can introduce the other parts of the plant, it is it's possible. It is already done. There is a technology for doing that, but we have to check the balance of the process. It is not the same working out with lignocellulosic material from palm trees, which is very recalcitrant if compared to sugarcane bagasse, for example. So the energy and the chemicals and the sustainability must be studied very carefully in that case.
All right. Just, I will repeat the question that you have replied now, because apparently there was a problem with my microphone. It was the question from Luis Ramos. He asked, could you use palm tree prunings, palm, fruit branches and residues to increase your yield in cellulosic ethanol? Can you tell us more about the technology used for this purpose? So this is the question you were replying right now. So just to remind the question, because there has been a problem with the sound.
Okay, don't worry. Here I go again. No problem about that.
So there is another question from Elena de la Fuente. She says, how do you fractionate to obtain the hemicelluloses without being degraded into microfibers? Sorry, to be integrated in the microfibers.
Okay, the hemicellulose is, in this case, galactoglucomannan. So we can dissolve them, we can dissolve and recrystallize them after. And it is very soft. That's a very soft process. We don't need very extreme and severe conditions. So it's very straightforward. We can do that very simply, and we can recrystallize the hemicellulose. I have to say that these sugars have some special behavior, so gives us the opportunity to recover all of them in a very pure way, in the shape of crystals or fibers, but we can also digest them further in the further process to obtain both single sugars or free sugars or oligos.
All right, thank you. So apparently there are no more questions. So probably we can leave it here. Thank you again, Javier, for this nice presentation, for such kind of replies that you have provided today to us, and it has been really nice to see other raw materials where we can obtain nanocellulose and the different applications you are working with, and also the the potentiality of its industrialization and the collaborations you have with the industry. So sorry again to the audience for the technical problems. It has been our first webinar, but apparently we have been able to solve it, and we have been able to ask to the speaker, and I hope we have enjoyed this first session of the webinar. So thank you very much, and I think that next month we will have another webinar, so I hope to see you all here. Thank you very much.
Thank you very much to you, Mark, for all your efforts. You were working very hard to do this possible, and I have an applause for you. Thank you.
Thank you.